Well, as part of this, which is our 100th birthday year, I have the distinct honor of welcoming you to our 2017 annual Edwards Lecture Series. And this year, it focuses on improving behavior through brain science. Today's lectures specifically focus on how studies of the brain enable us to better understand how we make sense of our complex sensory environment and then make decisions based on this sensory information. Now, before introducing this evening's lectures, let me just make a few comments about how this series came about. This truly exciting annual lecture series is a result of generous support of a former faculty member in our department, Professor Alan Edwards. And his family made a substantial gift to establish an endowment that ensures that these sorts of public lectures are made available to you every year free of charge. Professor Edwards was affiliated with our department for half of a century, from 1944 until he passed away in 1994. And he was an outstanding teacher, researcher, and writer who's credited with changing the way psychological science is conducted by introducing modern statistical techniques to this field. In fact, his statistical books had been the gold standard for psychological research for decades. Now, the Edwards family contribution to the psychology department is an example of what can be accomplished with the support from members of the community, such as each of you in this room, in fact. So please visit us at www.psych.uw.edu to see how you can be part of our future. And for those of you who have already contributed, and I know many of you are our friends, I want to thank you, and I applaud your contributions. So without further delay, let me introduce our first speaker for this evening. It's really with great enthusiasm that I introduce my colleague, Dr. David Gire. His broad interdisciplinary academic interest was evident early in his career as he double majored in neuroscience and philosophy at UC Berkeley. And then he pursued his interest in art and science at the International School for Advanced Studies in Trieste, Italy. It was only after that that he decided to go to graduate school to better understand human psychophysics, using a mouse model of sensory processing. And his pioneering graduate and postdoctoral work focused on understanding how the brain makes olfactory sense of our environment. And his work led to many high-profile publications that appeared in really the top journals, such as Nature Neuroscience, Neuron, and Journal of Neuroscience. Now, as you'll hear about this evening, his current work is trying to understand how our behaviors can regulate what we think we smell and how what we smell impacts our behavior. Now, this work is funded by a very prestigious NIH grant that's offered to new assistant professors, and I might say to just a handful of new assistant professors around the country. So really quite honored to have Dr. Gire here tonight. Thanks.
Well, thank you, Sherry, for that kind introduction. I'm really happy to be here tonight, and I'm glad that you all joined me. So let me tell you about an illusion I saw earlier today. In fact, it's an illusion that I think a lot of you have seen also. In fact, it's an illusion that we're seeing in this room right now. So what is this illusion, and what does this blurry black and white photo behind me have to do with it? Well, this blurry black and white image is what most of your eye is actually seeing when you look at the world. But it doesn't look like that to us. So why doesn't it look like that to us? Well, it turns out that in the center of our eye, we have this small patch that's jammed full of color photoreceptors called fovea. And we move our eyes around and scan the world around us with that fovea. And that's what creates the color vision and this high acuity vision that we think we have everywhere in our visual field, when in fact, what we're actually seeing is mostly a black and white blur. So if you collapse this over time, this is where somebody might be looking, where I might be looking if I were looking at the full resolution version of this painting. So to appreciate this a little bit better, let's do an experiment together. So the experiment we're going to do is I'm going to show you the high-resolution version of that painting, and I want you to figure out how wealthy that family is. Okay, so it's going to come up in just a second. Okay, look at the painting, figure out how much money those guys have. So ready? 3, 2, 1, go. Okay. So now we're going to do it again with a little bit of a variation. So this time, I want you to try to figure out how long the father's been away from his family. So ready? 3, 2, 1, and go. All right, so you may have experienced that when you looked at the painting for two different reasons, you were looking at different regions of the painting. And this was actually an experiment that was done in the 1960s by a scientist named Alfred Jarvis. And what he found was that people, when they were asked to look at the wealth of the family, looked in these regions here. So based on his eye tracking data, this is the only part of the painting that they would have been able to see in any kind of high acuity. The rest of it would have been, as you see here, a black and white blur. Meanwhile, if they're asked how long the father had been away, they looked mostly at the faces, maybe to see how excited the kids were that he was coming home. Now, if we project this data as colors onto the screen here, what you can see in blue is trying to tell how happy the family is to see him, how long he's been away, and in red you can see trying to tell how wealthy the family is. And what you can see is that there's very, very little overlap between those two distributions. So what this suggests to us is that when you're looking at this painting for two different reasons, you're actually seeing two different paintings. So now we're looking at it one last time in its full resolution. And despite what I just said about this illusion that we're all seeing, I'm sure all of us still think we're seeing this whole painting all at once in perfect clarity. But in fact, most of that's a black and white blur. So this illusion that I just told you about, it not only shapes our perception, but it also determines our decisions. And understanding it, how this happens, is the first big step that we can take towards improving the decisions that we make every day. All right, so what was the most important decision that you made today? I'm really hoping there's somebody in the audience who's thinking to come to this lecture, but regardless, I'm almost certain that nobody was thinking it was to look out the window at a tree at 3:15 p.m. We just don't think of these eye movements as being really important decisions to us in our daily lives. And there's a good reason for that. Our decisions span such a huge range of time, as you see here, from seconds all the way out to years. And these decisions about what to look at in our environment are very, very brief, and we make the decision in a split second. Meanwhile, there are other decisions we make, such as where to have lunch, which might take us seconds, minutes, or depending upon how picky you are, hours to make. And you might be taking things that you remember from your environment that you'd seen months or weeks before, using your memory to inform your decision. And then there are the big choices we make, such as where to go to college, where we might remember things over years to make that decision. It might take us weeks or months to actually make the choice. What we're seeing here is the decisions that we typically would consider to be important decisions. And these decisions are in these longer scales, larger scale decisions, and more longer term. Now in the lab, we're really good at studying these kind of short decisions, these decisions where we can take somebody into the lab and have them sit and do a task for a couple minutes and really well quantify things like where their eyes are looking and how quickly they might make a very simple choice. And now we know from what I was just telling you that these kinds of rapid sensory decisions feed into these larger scale decisions. And in turn, that these larger scale decisions come back and influence our smaller scale, more rapid decisions. So understanding this loop is going to be vital for us to understand how to improve our decisions that we make in our daily lives. And today I'm going to be telling you about how my lab is bringing these larger scale decisions into the laboratory environment so that we can start to understand them. So what's involved in one of these larger scale decisions? Well, you remember things. So for example, when you're going to find lunch, you'd remember where you'd had good food before. You're also going to sense things around you to try to help you, guide you to the thing that you want to find. So let's use finding lunch as an example. So here you see a map of the U District, with the university over in the right corner there. And the little red circle there is where I spend most of my time on campus. That's Guthrie Hall, the psychology building. So let's say you were leaving the psychology building and you wanted to go get lunch somewhere around the university. Well, where you choose to go would be very dependent upon how familiar you are with Seattle. So if you were just visiting from out of town, you might just have a vague idea that there's food over in the U District, and you just kind of wander around there until you find it. Meanwhile, if you were a local, you'd have all your favorite places already picked out, and you would just go right to where you remember they are. And now other factors come into play, such as proximity to Guthrie. So if you're feeling tired or lazy, you might decide you want to go to the closest place possible and try to remember which one that was. And then finally, a very rare event can occur in Seattle, and that's going to change what you're going to do. You're going to take advantage of that rare opportunity, and you're going to walk along the water until you find a place with a nice view. So how do we make these decisions, and how can we study them in the lab? Well, I think you see an obvious first problem with trying to study a decision like this in the lab, and that's something of the scale of the decision. So it turns out at the University of Washington, they don't give you a square mile of lab space when you start out as an assistant professor. So we had to come up with a different way to do this. And I'll give you a hint at what it was. So here you see 3 different proprietary pieces of technology and 3 equally amazed people using them. I'm not going to play favorites there. So what they're seeing is some form of a virtual environment. So that's basically trying to replicate what we're seeing every day in one of these devices so that we can see how people interact with their environment. Now let's explore that idea a little bit more before we jump right on board with it. So this is a close-up view of a Google Maps version of where we are right now. So that's Kane Hall, and over there's the quad. Now over the next couple weeks, we're all going to go out and enjoy the cherry blossoms. And I want you to think about what the experience of enjoying the cherry blossoms would be like. So you're walking among the trees, you're smelling the blossoms, you can hear the wind blowing through the trees, you can feel the wind on your face. If you choose to lay on the ground, you could put your hand on the ground and feel grass. If you decide to ignore all the signs that are everywhere, you'd climb a tree. Basically have a huge amount of freedom as far as what you're going to do in that environment. The same thing if you're walking to the fountain on a summer day and you feel the warm sun on your face, maybe smell some cut grass. So this brings up one of the big problems for us as scientists trying to handle virtual environments, and that's this enormous reduction in complexity. So we start out with this environment, which we're all living in right now, which has basically limitless complexity around us. And we're cramming it down into this tiny little box and trying to basically preserve as much of the important stuff as we can, while we have to discard a lot of other stuff. Now, for entertainment purposes, this is just fine. I'm happy playing these games if they're entertaining. I don't really care about the realism of the game. But as a scientist, I care a lot about the realism of the decision that people are going to be making in these environments. So to kind of tell you what we lose as we go from real to virtual, you have to really oversimplify the physics. That's unavoidable. And then movement is going to be altered. You're not going to be able to do everything you can in a real environment. So those are pretty much changed. But you gain something, which is control of the environment. And that's very, very valuable if you want to study decision-making in a really precise way. Now, can we have our cake and eat it too with this kind of approach? And I'm going to argue that with a little trick, we might be able to. So first, we're going to use something called hybrid virtual environments. Now, what I'm referring to here is we're actually going to build an environment in reality. So we get the physics for free, and the movement of the person or the animal in the environment would be for free. But then we run into a control challenge, which I'll address in just a minute. But first, I'm going to show you the little tweak we use to actually make this work in a lab. And there he is. So we actually use those little guys as our subjects. And now, why would we want to do that? Well, here you see the box that this little guy is going to be navigating in. to make his decisions. It's in red light because we're trying to simulate nocturnal conditions. It turns out these guys are nocturnal animals, and so they're really active at night. He's going to be basically asleep during the day. So that's why you're seeing red light, and you'll see red light through a lot of the rest of what you're going to see here. But anyway, he walks into this box, and to the mouse, what this box looks like is something like this. So it's this huge room. There's a lot of stuff we can do with it, and the decisions the mouse is going to make in the box can be these very complicated and kind of long-term decisions. Running to the opposite end of St. Peter's Basilica is not something you just do unless you're trying to exercise or something. A similar thing for the mouse. So there are real costs to making decisions in this box, basically in time and effort. Now, I know a lot of you in the audience are thinking, well, St. Peter's Basilica is very different from a box, and our brains are very different than the brains of a mouse. And this is almost to scale here, so you can fit a lot of mice in your brain. So your brain's that much bigger. So fortunately, through the decades, people have found that there are structural motifs in our brain, such as the hippocampus, which you see here, which show up also in the mouse brain. And through a lot of studies over the years, people have shown that these structures seem to be doing very similar things in the two types of brains. So we can discover some fundamental concepts in the mouse brain that are going to apply to some of the fundamental issues in our own brains. And finally, work by Sherry Mizumori's group and others recently has shown that mice retain many of our most prized cognitive abilities, such as impatience, greed, and laziness. And we call these things, we call them differently in science. So that's what we call them as scientists. So now let's take the control problem seriously. So we're going to put our mouse into this box, but how are we going to control the environment? Because that's going to be the difficult challenge now. We want to be able to place the lunch for the mouse wherever we would like in its virtual environment. All right, so what are we going to do? So here's our box for the mice and the rats to navigate. And now I feel like it's a little bit of magic goes on here, and that magic's going to be these two people here. So that's Brian Jackson and Sujin Ode. Brian's a grad student in the lab, and Sujin was a research technician. And what they did is they figured out a way to basically build a controller into this arena so that we can control the environment of the mouse or the rat with a computer and not have to involve humans interacting with the animals to place food or anything like that. And so they really took advantage of a lot of the resources on campus here. They spent a lot of time at Makerspace. They're working with the Institute for Neuroengineering, and they came up with this pretty cool idea, which was to take the hobbyist approach. So this is basically a DIY 3D printer. Those are Arduino microcontrollers there, and it's hooked up to the parts for a 3D printer. And what it's doing is, instead of printing something, it's dropping food throughout the arena. You can see it in action here. So there it goes. And so you can basically create any kind of distribution you'd like. You could drop the food pellets there. You could drop them in this other configuration. And then the really cool thing about this environment is that we are watching these rats who are doing this task for their entire life and their entire interaction with this environment. So we know everything they've ever done in that arena, and we know every place that they've ever found food in that arena. So this is going to allow us to really precisely test their memory and how they're using memory to make their decisions. So that's what you see here. We can use this system, and we can really get at that kind of, with this system, we can go weeks and months out into the life of a rat and actually understand how information and how memories are being integrated over such a long period of time. And that's because we have that precision of the hybrid virtual environment. But now there's another side to decision making, and that's the sensation, so our sensory input. So next we're going to look at that. So to do that, we're going to take a trip to a bakery with our mouse. So if you or I were to walk into this bakery and we forgot our phobias, it would look like that. And then if we got our phobias back, we'd be able to start scanning again. And we would put together a map like this probably in the first minute of looking at the bakery. And what you can see is we're looking at a lot of different kinds of croissants. We're looking at some signs, and we could probably figure it out with just this sparse information what we want to eat here. So what would our friend the mouse do in this situation? Well, this is as good as it gets for a mouse. So they can't do that trick that we use. But the mouse does have something that we don't, and something that Sherry alluded to in the introduction, and that's a sense of smell. They have a really, maybe thousands of times better sense of smell than we do. And so the world to them, this bakery anyway, would look something like this. So it would be a swirling kind of chaos of odor molecules that are being kind of buffeted around the room by turbulent airflow. So air is just kind of pushing them around all over the place, as you see right here. So as Sherry said, I come from an olfactory neuroscience background, and I come heavily from the neuroscience side of that. And so I was approaching this as a neuroscientist, and I was thinking, well, that's not so bad. I mean, they're swirling around. It looks kind of complicated, but it's nothing like the brain. So I was encouraged, too, because people had been thinking about turbulence in fluids since Da Vinci. And here you see a couple of drawings from Da Vinci where he was actually thinking about how turbulence might work. So it seemed promising. It seemed like we've had some hundreds of years of head start here, and I'm going to have a pretty easy time of it. So then I started reading some physics, and I ran into Richard Feynman, the Nobel Prize winner, who says that. So he says, turbulence is the most important unsolved problem of classical physics. Well, that seemed a little bit troubling. And then Horace Lamb, who another renowned physicist who famously said, I'm an old man now, and when I die and go to heaven, there are two matters on which I hope for enlightenment. One is quantum electrodynamics, which is what Feynman worked on, and the other is the turbulent motion of fluids. About the former, I am really rather optimistic. So at this point, I decided to collaborate. And the people you see here are two talented professors. That's Venkatesh Gopal, a professor of physics at Elmhurst College, and Agnesi Seminara, a professor at the CNRS in Nice, France. And working together with Venkatesh Agnesi, what we're hoping to do is to understand these complicated, kind of turbulent mixing kind of problems here and understand how that's going to create the sensory environment that our animals are running into with our superpowered noses. And so what you see here is an example of a simulation that Agnesi made. This is actually a three-dimensional simulation of a box like the one we saw earlier. And for those of you who aren't kind of acquainted with the problems in fluid dynamics, modeling this box took a cluster of supercomputers weeks. So this is not an easy problem. Turbulence actually turns out to be really, really hard. So, but Agnesi is able to get the code to work and to get these kinds of simulations out. And the big problem for the aficionados is we need to simulate down to very small scales, which is really hard with turbulence. So, but Agnesi got it to work, so we have these models. We need to figure out if the models work, if the models are giving us something real. So what we're going to do with Venkatesh's lab is they came out from Chicago, and we had kind of a build party with them, and we started building a bunch of laser-powered things in the lab, which is the way you should do science. Has to be lasers. And what these lasers allow us to do is to scan little bubbles. So we release these tiny bubbles into the air, and they're filled with a little bit of helium, just enough that they're neutral density with the air. So all we see is the motion of the air, not like the bubble rising or falling. And when you do that, you can actually see the turbulence in the air, sort of like how Da Vinci was imagining the turbulence in the water he was seeing. So now we're going to look at an experiment we did with the system, with part of the system that Venkatesh put together. So here we're actually just going to image a spray of, we're going to vaporize some water into the air, and we're going to use it as a proxy for where odors would go if they started at that little red dot there. Okay, so you can see it's the kind of blue flashing stuff there is when the water vapor hits the laser. We're scanning the laser basically throughout the whole arena. And so what we're seeing is where an odor cue would go if there were a food pellet right at that spot in the arena. And what you might notice about that stimulus, about this kind of pattern of odor coming off of that pellet, would be that it's very chaotic, it's intermittent, it doesn't look very easy to follow. And if we collapse it over time so we can figure out how reliable it is, we see that the red is really reliable, green is sort of reliable, and then the blue is falling off to nothing, and then purple's basically no signal at all. So what we saw from the physics is that within about half a meter, these animals could use this kind of sensory cue, but beyond that, it seems fairly hopeless as far as using it to do any kind of navigating towards their food. All right, so now we're going to look at an experiment. So we're actually going to put a rat into this arena, into this hybrid virtual environment that we've so meticulously built, and we're going to have the rat do its behavior, and its behavior is find lunch. So the lunch it's going to find are those red dots there, which correspond to the pellets that it's searching for. And what we'll superimpose on here are the odor cues that the animal might be experiencing. So we've estimated how far out the odor cue would extend, which is within one of those circles. And that tells us when the animal would hit kind of the estimated area where it could start using its senses to find these food pellets. And now remember, this is all being done under the red light, so they think it's nighttime and they can't really see anything anyway. And now this data was collected by Brian and then two talented undergraduates, Lulu and Sarahi, in the lab. So here comes our animal. Now this is sped up about 4.5 times, otherwise we'd be here all day. He's, as you can see, this animal's really not sure where anything is. Watching this video, it kind of reminded me of somebody who's in Seattle with an umbrella. So eventually the rat finds its food and its lunch in this new environment, but you can see it's pretty hard for it to navigate from any great distance. It's just kind of guessing. And then eventually it runs into the sensory cue there, and then it gets the food. And then it hears a tone after it's gotten all the food that tells it it's time to go back and go back in its home cage. So now we're going to go to fast forward to day 23 here. And the animals had time to be exposed to this kind of distribution of food pellets. So what you're seeing in yellow there is where the food pellets were falling with a really high probability for this rat. And blue were pellets that were much less probable. So we spent 23 days repeating this task over and over and over, and the rat really built up this kind of knowledge of where things should be in this virtual environment. And now you can think of the rat as having transitioned into local status, which in Seattle means he's wearing a Seahawks jersey. So he's really good at that now. And so one of the things we want to know is how that transition occurs. Because as you can see, we're quantifying what we just saw right here. The first days 1 through 10, the first sessions, the animal is finding about 20% in the first 2 meters of its search. By the end, it plateaus out at about 40% of the pellets within just the first couple meters of its search. All right, so now we can start talking about a kind of model we could build here to explain how we're integrating our senses and our memory to make these kinds of complex decisions. So early on, we saw that the rat was pretty much stuck using senses because it had that short-range olfactory cue, and it just had to wander around until it hit it. But then later on, it was able to build up a representation of the arena in its brain, and it used that information to find the food much, much more effectively. And now we can actually test this kind of model by tricking the animal a little bit. Now we're going to use the advantage of our hybrid virtual environment to drop a couple of pellets in really surprising locations. And those are shown with the kind of blue circles there. So these are basically in a location that the animal would never expect to find food after 30 days of training. But meanwhile, we have the red up there that are very expected. The animal certainly thinks that they'd be there. So let's see what the animal does. And again, here are the sensory cues. So you can see it does pretty much exactly that first trajectory, the very efficient one, but then we didn't play the tone, so it was wondering what went on, and then it comes back and finally finds the surprising pellets. So what this shows us is even really well-trained animals are using both strategies. They're not completely going over to memory. They're able to use both basically back and forth depending upon the demands of the task. And what we see here is we're going to quantify this a little bit. So these are the highly predictable pellet locations, and what we're going to show is a graph of how likely the animal is to move towards those pellet locations when it's doing the task. That's going to be shown on the y-axis. And then the x-axis is how far away the animal is from the pellet that it's trying to get. What you can see is when they're predictable, the animal's really good at doing this. It can start from over a meter away, even if we went off the edge of the graph there, and it goes almost 100% right at the food. So it's really able to navigate from far away to the expected locations. But then when we mix it up and throw in these unexpected locations, we see a significant decrease at long distances, suggesting that memory is really playing a strong role at the long distances. But then as the animal moves in closer, there's this real sharp increase, which tells us that the animal's hit that sensory cue and is now able to use its olfactory system to smell its way to the food. And now this question of how the animal uses the olfactory system to smell its way to food is something that's actually unsolved by anybody. We have 3 people in the lab working on it. I won't get a chance to tell you guys about their fascinating work today, but I just want to tell you Mike, Suzanne, and Debbie have been working on this, and they have some pretty cool results on it. All right, so how do we make these complex decisions based on what we just saw? So we have a memory aspect to making a decision like this, which gives us kind of this amorphous idea of where things should be in our environment. Now this might be, you had a really nice Thai meal over somewhere in the U District and somebody asks you where it was. You might be able to say, I don't know, it was over near 45th and University or something like that, and it started with a T. And but I really, I'd have to go back to help you find it, right? That's what you might say if you don't have a really sharp idea of where it is. And this is kind of the way memory's helping these animals, is it's putting them into the neighborhood of what they're looking for. That's shown here with that red line leading them to sort of close to what they want, but then their senses have to take them the rest of the way. So they get close enough with the memory and then switch over and start smelling their way to what they want to eat, and that's shown here. Now, what I'd really like to know is how this is happening in our brains when we're doing these complex decisions. And it turns out we had an opportunity to find out what's going on in human brain during a similar kind of decision. And this was through a collaboration in our department with Andrea Stocco's lab, and through Andrea with the GRID lab run by Jeff Ogeman. And then importantly, this was all spearheaded by Patrick Rice, a grad student in Andrea's lab, who took a neural dynamics course with me and said, I'd really like to study that kind of complicated decision-making in humans. So we worked together and we came up with a solution that I'll tell you now. So this solution is dependent upon something that's going to happen to 1 in 26 of us, and that's we'll develop epilepsy. So here you see a quote from Hugo Weaving, a famous epileptic, who says, I was diagnosed as epileptic when I was 13, and I had seizures once a year until my 40s. Then they just stopped. With every fit, my last thought always was, I'm dying. And then John Roberts, the Supreme Court, also has epilepsy. Both of these guys can't get a driver's license because of the danger of having a fit while they're driving, which could be fatal. So not everyone is as lucky as Hugo Weaving or John Roberts, and some people don't just have one seizure a year. They might have several seizures a week. They might have several seizures a day or more. And so we try to control these with drugs. Sometimes that doesn't work, and so neurosurgery would be required. And to do neurosurgery, what first needs to happen is the surgeon needs to really precisely map where these problems are coming from so that they only cut that one spot that's causing the seizures and leave everything else intact. So these patients who are making the data you are going to see possible are people who are kind of at the scariest part in their lives. They're going in for major brain surgery. And we're coming up to them and saying, hey, would you mind playing a video game for a while? And they said yes. So they're great. I mean, that's the only way we get this data, and it's only through the generosity of these people. So unfortunately, we can't bring our hybrid virtual environments to people in a hospital room because they're basically tethered to the machine that's recording, and they can't really leave the room, and it's really even hard for them to get out of a chair or out of the bed. So we had to go to a full virtual environment in this case. In this case, we'll be playing it. It's not going to be one of these headsets. We're still working out the problem of kind of getting that fit around the electrodes. Instead, this is going to be on a computer screen. So we're going to have this very reduced virtual environment. But thanks to our work with the rodents, we can figure out what part of the task the rodents were doing was important, and we can bring that into the human virtual environment, so we're studying the same kind of reasoning. And what I talked about earlier with the rodent was this combination of memory and sensation. We want to study that in people. So the memory aspect was actually pretty straightforward. We just made a virtual human arena that's basically the rat arena scaled up, so the human avatar here is kind of walking in a big warehouse. And then we can, of course, make the same distributions of the targets. In this case, this is just points that the person's getting for getting to the right spots. So that was pretty easy, getting the memory aspect in. The sensation was a little bit more tricky because the rodents were using this kind of local cue that was really highly fluctuating and chaotic and difficult to interpret. So with the humans, we took a visual approach because this is our main sensory modality. And what you can see is we made these swirling dots be the cue, and the person has to find them on the floor of this big warehouse, which is kind of covered in a mist. And the mist is there so that these are kind of enforced to be local cues, just like with our odors with the rodents. And now what we're going to do is see an example of a person navigating through one trial of this task. And what you're going to see up here is the data that we can collect at the same time. So this is the person's avatar up at the top. They're looking around. You can see as they're looking up, you see purple. They're looking straight ahead, it's green. If they look down, it's going to be blue. And making the arena like this allows us to see when they're looking down at the floor and trying to find something, because their trajectory is going to be blue. If they're looking up and trying to orient around the arena, it'll be green or purple. And then they're trying to get close to these targets, these yellow targets, which are marked with, again, those swirling dots. And so you can see this is basically how somebody would do this task. So next we decided to bring in a patient to try out the task. And so we don't get a lot of time with these patients. So this was actually kind of a race against the clock, because we want a task that's challenging, like that one, but we want the person to get good at it within about half an hour, because that's the amount of time we get with a patient. So they have to rapidly learn the task, but they also have to, it also should be challenging. And so we're trying to cut it really fine here. But fortunately, we had somebody who was really motivated. They said they actually really enjoyed the game, which was nice. And so they started out doing something like this. So they were mainly searching. You can see the blue is their trajectory as they're looking around. And they were mainly searching kind of close to where they started, which is that red square. Occasionally, they'd go out farther, but not often. And remember, the distribution of the targets was in those 3 locations there. But then after 20 minutes of playing the game really intently, they got much better at it. So now you can see that instead of clustering around this kind of early area here, they've pushed their search way out to the other side, and that's getting them more rewards. And we can do this statistics on it, and we see a significant increase in their performance. So the fraction of the targets that they find goes from 42 to 65% in only 20 minutes of playing the game. And that's great, because now we can actually study how the brain's changing during that time, because it's certainly making them do something different from this first part to the last part. So what's going on in the brain to do that? So first, if we want to understand what the data we're going to look at, we have to understand neural oscillations, because that's what the data looks like. So this is essentially what the data is going to look like coming out of someone's brain. So we have this electrode either on their brain or implanted into the brain. And what the electrode does is it picks up electrical activity of neurons that are around it. And neurons tend to do this kind of patterned oscillation. So you'll have this population of neurons just kind of rising and falling with their activity all at once. And this is the kind of thing we can pick up with an electrode. Now we record from many sites at once, so we don't just have the 2. And then the standard way to analyze this kind of data would be to see how well correlated those 2 sites are in their kinds of fluctuation, their oscillations. And so in this case, they're really correlated, really highly correlated. And you can see that we would have a big arrow or big connection between them. However, if the other site wasn't as well correlated, we'd have a small connection. And now the way we look at this kind of data is we use analysis techniques to kind of pick out the patterns that are going on in people's brains while they're doing different things. So next I'm going to show you real data from somebody doing, from the person that you just saw doing the task, who showed such a nice increase in performance. So here you see where the person was implanted. So they actually had electrodes right into their hippocampus, which is, again, that structure that's important, we think, for learning, memory, and spatial navigation. This is what a signal looks like coming from the 20 electrodes that were in the hippocampus. And now you can imagine that that activity is actually creating a map in the person's mind of their environment. That's what one of the things the hippocampus is thought to do. Meanwhile, we're also recording from the frontal cortex, shown in blue here. Again, 20 electrodes in the frontal cortex. And that area is thought to kind of guide our decisions, so guide cognitive decision-making kind of things. So in this case, like where to go in an environment or what to say to somebody. And now we have these two sites being recorded at the same time. So we can look at kind of this map-making site, and we can look at this kind of decision-making site, and we can ask how well correlated are they with each other while the person's doing this kind of very complex task. So the way we're going to display this, it's really hard to understand the data if you're just looking at it as kind of wavy lines. So I'm going to make this thing, which is not wavy, so maybe it's easier to understand, but I'm trying to compress the data into one structure here that we can all look at and understand the correlations between everything. And so what you're seeing at the top, or on the left here, is the hippocampus. That's in red, and every one of the electrodes is a little dot. On the right, in blue, is the frontal cortex. Every electrode's a little dot. Any line between a dot is a correlation. So that means the two sites were correlated with each other. And then a red line between red dots means the hippocampus was correlated with itself. Blue line between blue dots is frontal cortex correlated with itself. And then yellow lines are correlations between the two structures. Okay, and then finally, because I found it a lot more enjoyable to listen to music than to look at its waveforms, we're next going to actually run this through a little trick. We're going to pick 5 sites from the hippocampus, 5 sites from the frontal cortex, have them play the role of fingers on a keyboard, and whenever they exceed a certain threshold in oscillations, we'll have the finger essentially push a sound. So it'll sound sort of like somebody sort of playing a keyboard. Okay, and we'll be watching the person do, this is, again, is one of the first trials the person did on this task, so they're really new to this arena. So anyway, when I heard that, it sounded a little bit of a tune to it. I thought that was kind of cool that you could actually hear that in someone's brain. Made me think about, what's my brain doing all the time? What would that sound like? So next we looked at what happened when this person had then gone through all of this training for 20 minutes, and now they're very good at this behavior. And again, we're going to listen to their brain and look at the correlations over here. And now, just to show you what happened over that whole thing early in the training, these are all the correlations that were significant in the first few trials. So you can see there were massive amounts of correlations between frontal cortex and the hippocampus. So the decider and the map maker, essentially. Okay, so they're much more effective at finding the targets there. And when we looked at the correlations, we actually saw a huge decrease in correlations between our decider, the frontal cortex, and the map maker, the hippocampus. So if we look over the entire sequence of the training of this person, this is the first third, second third, and the third third of their training. You can see this gradual decrease in correlations between these two structures. But if we look at the correlations within the structures, there's no change. So it's really selective decrease in the correlation between the hippocampus and the frontal cortex. And what does this look like as far as the functional implications for decision making? Well, again, we have that decider in the frontal cortex, the map maker in the hippocampus. They start out really well correlated. And then they go to being not uncorrelated, but much less correlated than they were. And so what might this feel like to us? So I was trying to think about this, like, have I been in a similar situation? And the first thing I came up with was when I'm driving a car somewhere and my wife and kid are in the car and we're talking about something, if I don't know where I'm going or I'm trying to find something like a restaurant I've never been to, I just can't talk to them while I'm trying to look at the signs and go down this road. I can't split my concentration. I don't know if that's just me. But after I've been to the restaurant a few times, I can carry on a conversation just fine on the way driving over to that restaurant. And so a decrease in this kind of correlation between frontal cortex and a structure like hippocampus that might be making these kinds of maps might be something that frees up the frontal cortex to do other things. So this might allow the person in this case, while they're playing the computer game now, they could be talking to their parents in the room or something like that. So this is something we're going to be actively exploring as we move ahead, because there are ways we can test that hypothesis with these virtual environments. All right, so what I've shown you now is that we have this new way of looking at complex decisions in the lab. And we've been able to take it into humans and actually understand a little bit of what our brains are doing in these kinds of complicated situations. Now, next, we're going to take decision making. So I was talking to you about decision making on the scale from seconds to years. So let's extend that scale out to Hawaii. So 500 million years of decisions. So that's the thing we'll study next. How are we going to do that? Well, it turns out nature ran an experiment for us, and we just showed up now to pick up the results. So right now, what you're seeing here is a tree of life, a very kind of truncated tree of life, but we're going to look at the relative locations of the rats, the mice, and us. We're all kind of clustered in that one spot in the tree of life. But there's another animal out there which literally has been making 500 million years of different decisions than we have, because the last common ancestor we had with an octopus was about 500 million years ago. So to bring that home, this is the last common ancestor we have with rodents. It kind of looks like a rodent. It has a lot of our same body plans, such as a centralized brain, bones, stuff like that. So if our form of decision making is similar to that of the rodents, it might just be an accident of evolution. It might not be the best way to make decisions. So the octopus gives us a chance, as much as possible as we can with life on Earth, to test the hypothesis that there might be another way to make decisions. So here you see a picture of an octopus. It does have a centralized brain, just like we do. Its brain's actually wrapped around its esophagus. But in addition to that centralized brain, it has this second brain, which is actually larger than its first brain, which are the arms. So it has these ganglia going out in its arms that actually do a lot more thinking than the brain in the center there. So this is completely different than our body architecture. And we're now wondering, how are these guys using this different brain to make decisions? Now, to do that, we're collaborating with another group in psychology, Joe Cisneros' group. And again, this is a project that was completely driven by a very talented graduate student, Dominic Civitilli, who you see here in his natural habitat. And he's out there with his dive team in the Puget Sound. And then an undergraduate, Susan, who's been working with Dominic to help maintain the octopuses. So now you're going to look down into one of the tanks in which the octopus lives, and we're starting to build these kinds of rudimentary hybrid virtual environments for the octopus now. So this is one of our first simple tasks, which we called rock, clam, mussel. So the octopus has to tell the difference between a rock, a clam, and a mussel. And octopus is going to come on screen from the edge here and try to make that decision. So here you see it kind of walking along the edge. It's gone to one of the objects, and now it's really going to start the decision making. And now you see that second brain is actually kind of spread out through the environment, and the animal's using its arms to sense what's in the environment. Now, importantly, in this situation, we're also operating under red light to mimic nocturnal conditions, because that's when octopuses also hunt. And the way they hunt is just like the mouse, they're using odor cues. And they're picking up those odor cues with their arms. It's actually using the suckers in their arms. So as we watch the arm move around the environment, we're actually seeing them reach out and try to grab odors and smell them. So this allows us to use computational techniques borrowed from computer vision to trace the arms of the octopus here each individually. And we can start seeing what kind of algorithms, what kind of strategies the octopus is using with this distributed system. So it's still early days for this project. I think it's quite exciting, and we're just going to see where the data leads us with this. But I think we might see some very big and interesting differences here. All right, so I'm getting the 2-minute warning here, so I'm not going to belabor what I've shown you today. We talked about the decision making. We talked about we're doing this in human brains, and we talked about the fact that we're looking at this in a creature that's 500 million years distant from us. Now, I need to make a really important point here. Well, one reason is some legislation that was passed in Britain today, or the letter that was sent from Britain to the EU today. I want to make a point here that we need to emphasize that the results you saw today came from a team effort involving people from 8 different countries. And these people grew up speaking several different languages. Our success that we have is a direct result of working both across scientific borders as well as across national borders. And this is our research team who you saw earlier throughout the talk. Now, another point I really need to emphasize here is that today you've heard about work from a group of people from a variety of scientific disciplines, and everyone had one common goal. And that goal was to understand decision-making in the real world. And this combination of science across disciplines, unified behind a focused question, is only possible in a department that both values scientific diversity as well as encourages collaboration across that diversity. And that is the Department of Psychology here at the University of Washington. Importantly, the projects you have heard about today were all started and are currently directed by graduate students. This impressive constellation of collaborations you see here is only possible because of their efforts. And finally, if some of you out there, especially undergrads at UW, would like to come and work with us, just drop us a line at sense.decide@gmail, and we'd be happy to talk to you about it. And there are a lot of other projects we've been working on. I can't tell you about all of them today or be here forever. But I really want to thank everybody who's been helping my lab in the first couple of years we've been here. It's just been an incredible environment, and I'm really lucky to be at the University of Washington. So thank you.
Great. Well, we have a couple of minutes for questions, and then while people are thinking about who wants to come to the microphones, after these couple of minutes, we're going to take a short break while we transition to the next speaker. And during that time, you also have an opportunity to come down and talk to David. Okay? Yes, there's one question right out here.
So what are the most exciting or intriguing applications of what you're seeing so far that you can imagine making a difference in the world?
What are the implications?
Applications.
Of the applications. So that's actually leading up to our next talk. So Raj is going to show you some really cool stuff that they're doing with the data you saw from the humans here. And there's a little preview there in Andrea Stocco's photo about how you can make brains communicate with each other, which is going to come up in the talk. But I think from the decision-making side, we just make so many of our decisions without thinking about it. It's because these decisions are kind of these hidden decisions. So if you walk into a room, your eyes are scanning, and you're scanning at the things that you're kind of predisposed to look at as you walk into that room. You're doing this unconsciously and without intention. And maybe if, let alone putting electrodes in people's brains or recovering movement and paralysis, but if we just face the world with a little bit more intention and maybe brought the frontal cortex into things a little bit more, we might be able to change what we do without having to put electrodes in our brains. That's what looking at this kind of unconscious processing has really been bringing home to me.
Thanks.
We have time for maybe one more question, or? Yeah.
I'm wondering if the School of Business Marketing Department has any use for this in some of the behavioral targeting or
Yes. Yes, they certainly do.
I don't know if people, I don't know if they
Oh,
heard the
yeah,
question.
sorry. The question was, would business have a marketing use for this kind of thing, like School of Business? And yeah, that's a big field, actually. That's
We should talk.
yeah, exactly. Hey, if somebody, but no, they actually use. So we don't use eye trackers here for technical reasons with the patients, mainly because we don't have enough time to calibrate them. But business people use eye trackers. There's this whole kind of neuroscience of economics that people look at, where you can actually show people ads and track their eyes. And yeah, I'm a little bit disturbed about the implications there, because I think you can really hijack our attention. And if you hijack it enough, you get people looking at the wrong things enough, you might actually change the decisions we make, right? I mean, that's the point of advertising.
Yeah.
Yeah.
Thank you.
Sure.
Thank you.